Welcome to Chapter 4, Lecture 3. We are going to continue on with our lectures. You should have gotten this note packet from the substitute today, and we're going to be talking about eukaryotic cells in this lecture. So let's talk about some learning objectives. Things that you need to bring out of this lecture are you need to be able to name examples of eukaryotic cells. This one's actually pretty easy. Just name you and me. We're eukaryotic organisms. Okay, you need to be able to compare and contrast prokaryotic and eukaryotic cells, aka the outside circles of our Venn diagram on our worksheet. And you need to be able to identify and explain the different parts of a eukaryotic cell. Then finally, we're going to start talking about the differences between the two main types of eukaryotic cells, plant and animal cells. So we need to know what are different between these two types of cells. Okay, so this is a schematic of a eukaryotic cell. Now, in general, eukaryotic cells get considerably larger than prokaryotic cells, right? There are several examples of eukaryotic cells that we've talked about in class that we can see with our naked eye. Okay, now, eukaryotic cells differ from prokaryotic cells in the fact that they have a nucleus, this guy right here. The nucleus is where we store our genetic material, aka our DNA. But what we also have are these membrane-bound organelles. Membranes are actually really important in how a eukaryotic cell functions. Okay, these membranes are going to be like different compartments. Okay, you can think of them like the apartment analogy that we've been talking about. So eukaryotic cells are the two-bedroom apartment, which means it has separated living room, separated kitchen, and separating sleeping quarters for whoever's living in that apartment. These different rooms are going to allow for different chemistry to happen. So different things are going to happen in different organelles and different compartments based on what's needed by the cell. Now, just like prokaryotic cells, there are some things that we have in common, right? We have our DNA, our genetic material, is just found in the nucleus instead of the nucleoid here. We are going to have cytoplasm. We're going to have ribosomes. These are what the little red dots are. These are our ribosomes. Okay, and then we are going to have the cell membrane. Okay, that boundary that keeps the outside separated from the inside of the cell. So let's talk about some of these structures in a little bit more detail. Okay, so as we start going through the different parts of the cell, the first one we have to talk about is the one that is similar for all cells, right? All cells have plasma or cell membranes. Plasma membrane, cell membrane, different words for the same thing. Okay, both prokaryotes and eukaryotes have a phospholipid bilayer that's going to make up the plasma membrane. But what's found in the plasma membrane is a little bit different between the two. So the eukaryotic plasma membrane is slightly different than the prokaryotic plasma membrane, even though they both have one. So in eukaryotes, we're going to have embedded proteins. So will prokaryotic cells. They're just going to be slightly different. Now, these proteins are really important because they're going to help with what's called transport, and they're going to help with cell structure and cell stability. Okay, transport-wise, some of these proteins are going to allow things to come in and out of the cell. Okay, it's going to allow for the passage of things like water and glucose and amino acids, and really importantly, ions. Some things like calcium 2 plus and sodium 1 plus and chlorine 1 minus. Okay, we're also going to transport oxygen in and out of the cell. Okay, these are all needed for the cell to be happy and healthy. Now, one of the things that's different between eukaryotic cells and prokaryotic cells is some of the other things that are found in the plasma membrane. Okay, one of the biggest differences is that eukaryotic plasma membranes have cholesterol in them. Okay, this cholesterol is really important because it's going to help our membrane maintain something called its fluidity. So how fluid and how flexible is it going to be? Okay, this is going to be something that's going to help in different temperatures. If a eukaryotic cell is in really cold temperatures, without cholesterol, it'd get rigid and really solid and wouldn't be able to bring things in and out. Conversely, if it's in really hot environments, our plasma membrane is made from phospholipids, right? If we heat up a lipid, it's going to get very liquid. Okay, we don't want to liquefy our cell membranes, right? That's that boundary that's going to keep the inside and outside separated. Okay, cholesterol is going to help in the hot temperatures to keep the cell membrane together. We'll talk more about this in chapter 5. Okay, other important things is some of these proteins are going to allow things like carbon dioxide and ammonia to escape, right? One of the characteristics of humans is that we breathe oxygen gas in, we expel carbon dioxide. Well, carbon dioxide is made in our cells, so it's a waste product. Too much carbon dioxide in a cell can actually cause it to become to have an acidic pH, so it's going to become more acidic, right? This is how we make soda carbonated, is we bubble CO2 through it, which also makes soda slightly acidic. Okay, so we're going to allow waste products like carbon dioxide and ammonia to travel through the membrane as well. So the second cell part we're going to talk about is the cytoplasm. Now, all cells have cytoplasm, right? This is one of the four things that all cells have in common. But in eukaryotic cells, it actually defines a specific region of the cell. Because eukaryotic cells have a nucleus, we say that the cytoplasm makes up the region between the nucleus or the nuclear envelope, so where the nucleus is found, and the plasma or cell membrane. Okay, so this area is where the cytoplasm is going to be found. There is no cytoplasm in the nucleus. And this is important to remember. We have something else in the nucleus, and we'll talk about that in a couple slides. Now, our cytoplasm is made up of something called cytosol, and about 70 to 80% of it is water besides that. Now, there's a lot of things dissolved in our cytoplasm. We're going to have things like glucose and proteins and ions like calcium 2 plus and sodium 1 plus and chlorine 1 minus. And we're going to have amino acids. Okay, but what we're also going to have is a lot of proteins, right? All those proteins that the cell needs in order to function correctly are going to be found in the cytoplasm as well. Okay, in fact, even though the cytoplasm is 70 to 80% water, the number of proteins we have dissolved in it is going to give it that gel-like consistency. Okay, so it's going to be semi-solid. Now, the other thing that our cytoplasm holds is the cell organelles. Okay, all of our cell organelles are suspended in the cytoplasm. So the next part of the cell that we're going to talk about is something called the nuclear envelope. Okay, the nuclear envelope is going to protect our nucleus. Okay, so you can kind of think about it like the cell membrane, but for the nucleus. So it's going to keep the nucleus separated from the cytoplasm. So our nuclear envelope is a double membrane. Okay, it's going to separate the DNA from the cytoplasm, just like the cell membrane separates the inside of the cell from the outside. Okay, but it also has another function. Okay, it's going to separate the two parts of the central dogma of biology, right? The central dogma of biology states that information flows from DNA to RNA to proteins. Now, each of these processes has its own name. Okay, so when we're going from DNA to RNA, this is what we call transcription. Transcription happens inside the nucleus, inside the nuclear envelope. Okay, whereas the second part, when we go from RNA to proteins, this is what we call translation. Okay, for translation to happen, it needs to be in the cytoplasm where all those amino acids are and where the ribosomes are so that the ribosomes can make proteins from that RNA. So one of the functions of the nuclear envelope is to protect our genome, to keep things out that would break it down, and to separate transcription and translation from each other. Now, our nuclear envelopes have proteins in it called nuclear pores. Okay, our nuclear pores are these kind of raised options here. Okay, these are going to be scattered throughout the membrane, and they're going to perforate, or they're going to cross both the inner and outer membrane, so this double layer. Okay, and just before I start talking more, there is a space in between the outer and inner membrane, and we call this the perinuclear space. Okay, so our nuclear pore complex, these are going to allow for things to come in and out of the nucleus. Okay, but they're going to be really picky about what comes in and out. So they're going to regulate not only the flow of molecules back and forth, but they're going to regulate what's in the cytoplasm. Okay, because there's a lot of stuff in our cytoplasm that we don't want to get into our nucleus with our DNA because it's going to cause it to get damaged. Okay. In fact, the only way that you can get in through the nuclear pores is by having what is called the nuclear localization signal pass through. Okay, so there's certain molecules that are going to open these pores and some that are going to keep them closed. Okay, so if you want to gain access to the nucleus, you have to have a specific type of molecule to bind to these pores that's going to open it and allow things in and out. Now, our ribosomes are also made inside the nuclear envelope, so they have to be able to come through as well to get into the cytoplasm so they can make proteins. So inside our nuclear envelope is the nucleus. So if we look at our really simple diagram here, we have our nuclear envelope. We can see that here, and we can see our nuclear pores, which are going to allow things to go in and out. But then inside of it, we have our nucleus. Inside the nucleus, we have the nucleoplasm, and we have the nucleolus, which we'll talk about in a couple of slides. But most importantly, inside the nucleus is the DNA. We sometimes refer to this as the chromatin. Chromatin and DNA are the same thing. Okay, that's the job of the nucleus, is to store our DNA, but it's also where our DNA gets copied or replicated. So we talked about the surface area to volume ratio problem that cells have in lecture 2. If a cell gets too big, like it has too much volume and its surface area can't support that volume anymore, our cells have one of two options. They can either divide or they can die. Okay, a lot of cells are going to choose to divide instead of to die. Now, one of the prerequisites before a cell can divide is a cell has to have 2 copies of its DNA, one for each of the new daughter cells that are going to be present after the cell divides. So that replication process, that process of copying the DNA and making 2 copies, happens in the nucleus. Another process that happens in the nucleus is transcription. Okay, that first part of the central dogma where we go from DNA to RNA, that process also happens in the nucleus because we want to keep our DNA in the nucleus if at all possible. Now, all cells are going to have at least one nucleus. There are some cells that have 2 nuclei, and there's some that accidentally get 2 nuclei, but those are going to be exceptions, and we're not really going to cover those in this course. Okay, so right now we're just going to assume every cell that we look at that's eukaryotic is going to have one nucleus. Now, the nucleus is also going to be the largest organelle found in the eukaryotic cell. In fact, it's so large, we can see it underneath the microscope very, very easily. We have special stains that are going to highlight the nucleus and help us see it even more. In fact, one of the first labs that we do, we're going to look at different types of cells, and we're going to stain them ourselves in lab, and we're going to look at the nuclei that we can see in there. Okay, the nucleus, as I said, it's a large organelle. To put it into perspective, it is larger than most prokaryotic cells. Okay, so it's exceedingly large, right? It's bigger than the mitochondria, and the mitochondria has about the same 1 micrometer size as most prokaryotes. Now, besides storing the DNA and helping with DNA replication, the nucleus is also really important because this is where we make the ribosomes. We make the ribosomes in the nucleolus. Okay, so it's the way of the nucleus to direct the production of proteins. Now, all of our information that we need to make the proteins our cell needs in order to function is found in the genetic code in our DNA. Okay, but the ribosomes are what's going to actually take that information that we have stored in the DNA. We're going to put it into RNA, and then the ribosomes are going to read the RNA to make the proteins. Okay, so all of that happens because of the nucleus. So we need to pause for just a second, and we need to talk about DNA specifically. We need to talk about some vocabulary that we have that goes along with DNA. And these vocabulary words are going to describe different time points for the DNA. Now, we have our DNA double helix, right? This is going to be our 2 strands that are formed from the hydrogen bonding between the 4 bases, right? DNA is made of nucleotides of adenine, guanine, cytosine, thymine. Okay, this is going to give us our double helix. This is the base structure of DNA. But what we do with that DNA after we have the double helix is going to lead to the other terms. Okay, our other terms are chromatin. This is the state that DNA is in most of the time. And then we have the chromosomes. Okay, now chromosomes, these are the structures in the nucleus that are made of tightly wound or supercoiled DNA. Okay, a lot of times when we're talking about DNA and we're talking about genetics, we're talking about the chromosomes. Okay, each organism has a characteristic number of chromosomes. Okay, in prokaryotes, the DNA is tightly organized and tightly wound into one single circular chromosome. In eukaryotes, the chromosomes are linear. In fact, after replication, they kind of take on this X-like structure. Okay, in fact, we can see DNA in the cell when it is supercoiled into chromosomes. Okay, so every eukaryotic organism is going to have a unique and specific number of chromosomes in the nucleus. So humans have 23 pairs, or 46 chromosomes in each of our cells. Other organisms can have more or less. Okay, plants tend to have more chromosomes than humans, and things like bats can have less. Okay, but chromosomes are these supercoiled structures. Okay, we only really see chromosomes when a cell's getting ready to divide. Okay, so then we're going to see the two sets. DNA is mostly found as chromatin. In fact, a lot of times when we're looking at different labeling systems and different structures and different diagrams when we're talking about the cells, instead of seeing what's in the nucleus labeled as DNA, we're going to actually see it labeled as chromatin. Because when the cell is not dividing, our DNA is not going to be supercoiled into chromosomes. It's going to be found in chromatin. Okay, so chromatin is where we take our double helix DNA and we wrap it around these proteins called histones. Okay, sometimes we call it beads on a string because of how the DNA winds around these proteins. Okay, so this is what chromatin is. Okay, this is going to also allow us to fit all of the DNA into the nucleus of the cell, even though we have more DNA that should fit if we talk about how long DNA is inside of the cell. Okay, so we're going to have this loosely wound beads on a string. Okay, this is what's going to be found in the cell at all times. Okay, in fact, chromatin is the word we use to describe the DNA when it's both condensed, okay, aka found in the chromosome, and when it is decondensed. So when we're trying to unwind it from these protein histones in order to open up the DNA so we can copy it. Okay, which then brings us to the last two parts that make up the nucleus. Okay, so we have the nucleus, it has the chromatin inside, but in the center of the nucleus is kind of like its own nucleus. It's called the nucleolus. Okay, this nucleolus region is actually really important. This nucleolus is where we make ribosomes. Okay, so we have a specific type of RNA called ribosomal RNA, and we have different proteins that are going to build these ribosomes. Okay, well, once the ribosomes are built in the nucleolus, we're going to ship them out through the nucleoplasm and through the nuclear envelope into the cytoplasm. Okay, so the nucleoplasm is the cytoplasm of the nucleus. Okay, it's going to be semi-solid like the cytoplasm. And this is where we're going to find the chromatin. We're not going to find much else besides the chromatin here because we don't want anything like we have in the cytoplasm, because some of the stuff in the cytoplasm will literally degrade or break down our DNA. Okay, so the nucleoplasm is going to protect our DNA as well as the nuclear envelope. Okay, so let's talk about our ribosomes. These are the fourth thing that all cells have in common, right? All cells have DNA, all cells have a cell membrane, all cells have cytoplasm, which brings us to ribosomes, the last thing all cells have in common. Now, ribosomes are interesting because they're going to participate in making proteins. In fact, they are the ones that build the proteins. Okay, ribosomes are made of what we call two subunits, so they are actually two proteins put together. We have the big subunit. Okay, this is actually literally called the 50S subunit. And then we have the small ribosome subunit, which we would call the 30S subunit. Okay, so both prokaryotes and eukaryotes have ribosomes. The ribosomes for eukaryotes are slightly bigger than that for the prokaryotes. Okay, size does matter here. Okay, and our ribosomes are made up of proteins and a special RNA called rRNA or ribosomal RNA. Okay, so when these two subunits come together, they form the ribosome complex. The ribosome can read our RNA called mRNA, and the mRNA is going to take the information from our DNA about how to make our proteins and make it so that the ribosome can actually read it and build a protein from it. Okay, so all the information for what amino acids go into our proteins is found in the mRNA. We're going to talk about the ribosome and this whole process of translation, because the ribosome does translation, aka making the proteins in the central dogma. So we're going to talk about this in more detail in the second semester. Okay, so our next part of the eukaryotic cell that we're going to talk about is the one that we all know after 7th grade life science, and that is the mitochondria, aka the powerhouse of the cell. Okay, the mitochondria has a really important job. This is where the energy is made that our cells can use, aka ATP. ATP is the cellular currency of energy, meaning this is the form of energy our cells can use directly. Okay, we get energy from carbohydrates, we get energy from glucose, we get energy from starch, we get energy from glycogen, but we can't use it directly. So what the mitochondria does is it takes those carbohydrates, breaks them down to glucose, and then takes that glucose and then further breaks it down into a usable form called ATP. Now, mitochondria are really interesting. They are a large organelle structure. In fact, mitochondria are as big as most prokaryotic cells. There's a reason for this. So we think that eukaryotic cells obtained our mitochondria via a process called endosymbiosis. Endosymbiosis means that our ancestors to our modern eukaryotic cells ate a prokaryotic cell back in the day. Instead of breaking that prokaryotic cell down into nutrients and things that the cell needed, it struck up a relationship with this prokaryotic cell. It's called a symbiotic relationship. So symbiotic relationships mean mutually beneficial, meaning that the eukaryotic cell provided a place for the prokaryotic cell to live, and the prokaryotic cell provided something for the eukaryotic cell. In the case of mitochondria, it makes energy for the eukaryotic cell and does very well at making this energy. Now, how do we know that the mitochondria is a prokaryotic cell that our eukaryotic cells ate back in the day? One of the pieces of evidence we have for this, and we'll talk about this on and off as we go through the next few chapters, is that mitochondria have their own DNA. No other cell organelle besides chloroplasts, we'll talk about those when we talk about plant cells, have their own DNA. Okay, not only do mitochondria have their own DNA, that DNA is, for the most part, passed down from your maternal line. Now, we do have some evidence that this is not always true, but it's true a lot of the time. So the DNA in your mitochondria could be the same as your mom's, which could be the same as your grandma's, which is kind of cool. But the shape of the DNA is actually really important because eukaryotic cells have linear chromosomes, right? Prokaryotic cells have circular chromosomes. Mitochondria, the DNA that they have, is circular like prokaryotes, which makes us think that mitochondria used to be a free-living prokaryotic cell back in the day before it got ate by our modern eukaryotic cells. Okay, but mitochondria, they're going to make energy, and part of the reason why they can do this is the fact that the mitochondria has two membranes. Okay, so it has an outer membrane that looks very much like a eukaryotic cell membrane, and it has an inner membrane that looks like a prokaryotic cell membrane. Second piece of evidence that our current eukaryotic cells' ancestors ate a prokaryotic cell. Okay, this inner membrane is folded. Okay, the folds are called the cristae. This inner membrane is what is going to convert glucose into ATP. Okay, and it's going to use this intermembrane space in between the inner and outer membranes to do that. Okay, now inside the inner membrane is what we call the mitochondrial matrix. Okay, so it's this fluid that's going to fill in between the cristae and between the folds. As I said, we'll talk about endosymbiosis in more detail as we go into a couple more lectures, but kind of cool. Okay, the next organelle that we're going to talk about is the peroxisome. Okay, peroxisome, that's a fun name. So peroxisomes are a small round organelle with a single membrane. Okay, so mitochondria has two membranes, peroxisome only has one. So inside this single membrane is what we call the crystalline core. So this crystalline core is made out of peroxides. So peroxides are very damaging to a lot of molecules. So this crystalline core can break things down. Specifically, it's used to break down fatty acids from lipids, and it breaks down proteins into amino acids. Okay, another function that the peroxisome has is it's our first line of detox when we run into things that are poisonous for our cells. Okay, we can take up the poison into the peroxisome, and those peroxides, just like they break down fatty acids and proteins, they can break down poisons so that they're no longer causing damage to our cells anymore. Okay, so this leads us to a large set of organelles that are interconnected. We're going to call it the endomembrane system. Okay, so the endomembrane system are those membrane-bound organelles. So these different compartments are going to do different chemistry, which allows for the eukaryotic cell to survive and get larger than a prokaryotic cell. Okay, so the endomembrane system consists of a lot of internal membranes. Okay, so membrane here, all the membranes. Okay, including the plasma membrane. Okay, so we're going to have these different internal membranes. They're going to have organelles suspended in them, right, membrane-bound organelles. And these organelles are going to work together to modify, package, and transport both lipids and proteins. That's their job. Now, the endomembrane system is going to consist of the nuclear envelope, which we've talked about, an organelle called lysosomes, which we'll talk about on the next slide, vesicles, the endoplasmic reticulum, which is fun to say, and something called the Golgi apparatus. Also, the plasma membrane is included in the endomembrane system, but we've already talked about that. So the first organelle that we're going to talk about in the endomembrane system is the lysosome. Lysosomes are special. One, they are only found in animal cells, which means they're not found in plant cells, they're not found in prokaryotes, they're only found in animal cells. So they're a defining characteristic. In fact, they're one way that we can tell animal cells from plant cells, besides the plant cell wall. Okay, lysosomes are membrane-bound organelles that contain digestive enzymes. They are what I like to think about as the garbage disposal of the cell. They break everything down that comes into the cell. Okay, they can break down large biomolecules, they can break down worn out and excess organelles, and they even break down invading viruses and bacteria that enter the cell to cause infection. So if we go through this little diagram here, this is a cell. This is probably amoeba because it has a pseudopod or fake foot. Okay, so our little amoeba here is going to eat a food particle. It's going to bring it in a process called phagocytosis, which we'll talk about in chapter 5. Okay, so it brings this food particle in. It's going to encapsulate it in a membrane called a food vacuole. Okay, so it's going to come into the cell, and then our lysosome, containing its digestive enzymes, is going to come and fuse with this food vacuole. Okay, we call this the endosome lysosome complex. Okay, and once it fuses, all those digestive enzymes have free access to the food particle, and it's going to start to break it down. Okay, and then it's going to recycle those parts of the organelle or the macromolecules that it broke down, and the cell's going to then use them. And sometimes it just gets it out of the cell. Okay, now lysosomes also participate in a process called apoptosis. When cells get damaged beyond repair and they can't fix themselves, lysosomes are going to help the cell self-destruct. Okay, this is what we call programmed cell death or apoptosis. We'll talk more about it when we talk about cell signaling. The next part of the endomembrane system we're going to talk about is the endoplasmic reticulum, or the ER. We're going to start referring to it as the ER because saying endoplasmic reticulum over and over and over again, it gets actually pretty hard. In fact, try to say it 5 times fast. Now, the endoplasmic reticulum is a network of interconnected membrane sacs and tubules. Okay, so these are membrane sacs and tubules. Okay, the endoplasmic ER has 2 parts. We have the rough endoplasmic reticulum and we have the smooth endoplasmic reticulum. Each has its own job. Okay, the rough ER, or the rough endoplasmic reticulum, its job is to make and modify proteins, and the smooth endoplasmic reticulum, its job is to make lipids. Okay, so this is where a lot of the things that our cell needs to function is made, is in the endoplasmic reticulum. So the ER is made of these membrane sacs and these tubules. These tubules are actually hollow. Okay, so the hollow part is what we call the lumen or the cisternal space. Now, what's really important to remember about the ER is that it's continuous with the nuclear envelope. What does this mean? It means that the nuclear envelope that holds the nucleus is literally connected to the endoplasmic reticulum. It's one big network of membranes and tubules. Okay, so they're connected to each other. Okay, so let's keep talking about the endoplasmic reticulum, but let's divide it up into a different section. So we're going to talk first about the RER, or rough endoplasmic reticulum, and then we'll come back to the smooth endoplasmic reticulum, or the SER. Now, the RER is named because when you look at it underneath the microscope, it looks like it has a rough surface. And the reason for this is where we find a lot of ribosomes that aren't just free floating in the cytoplasm. Okay, the ribosomes are going to be attached to the membranes in the RER, and this is going to give it that rough look that it has. Now, I would say that the rough endoplasmic reticulum is the workhorse of the cell. Okay, because this is where most of the ribosomes are found, this is where a lot of the proteins are made. In fact, this is the RER's main function, is to make proteins. Okay, so our ribosomes are going to make those proteins. Okay, they're going to go through and fold here. And we can also modify our proteins here, which means that we can add side chains, we can cut pieces off, we can do things that are going to allow the protein to be fully functional. Okay, all of the modification happens in the lumen of the ER, in that cisternal space. Now, modified proteins are either going to be incorporated into the cell membranes, or they're going to be released from the cell. Okay, so think things like protein hormones like insulin, and things like enzymes that are needed to break things down or build things up. The main job with RER is to make and modify proteins, but the RER also makes phospholipids for the cell membrane. So phospholipids and a lot of these modified proteins aren't destined to stay in the endoplasmic reticulum. They actually need to be shipped where they are most needed. And sometimes they need to be shipped out of the cell completely. So one of the things that the RER does is it packages these things to get to where they need to be. And one of the ways it does this is by putting them into something called a transport vesicle. Now, the RER is going to take part of its membrane and it's going to encapsulate the protein or the phospholipid. Okay, so if we look here in our picture, we see the membrane sacs and tubules that make up the RER. Okay, we see all of our little ribosomes embedded in the membrane. But what's going to happen is, say we have this protein and it's ready to be exported. Like, we've made it, we've modified it, and we need to get it where it needs to go. What's going to happen is we're going to take part of the membrane from the RER and we're going to surround that protein. Okay, this is what's called a transport vesicle. It's going to transport it to where it's needed. Now, a lot of times these proteins need to go to the Golgi apparatus first. Some things can go directly to where they're needed, or they can go straight, if it's a phospholipid, straight to the plasma membrane if it doesn't need modifications. Okay, but this transport vesicle is going to make sure it gets to where it needs to go. Okay, so once it surrounds the membrane around the protein, that vesicle that membrane is going to bud off to make the transport vesicle. So we've talked about the RER and the fact that it makes proteins and phospholipids. We now need to talk about the smooth endoplasmic reticulum, or the SER. So the SER is attached to the RER. It's continuous, and it's continuous with the nuclear envelope as well. But one of the biggest differences between the SER and the RER, and one of the reasons why the SER is smooth, is that there are no ribosomes in the SER. Okay, there's nothing there to give it that rough appearance. Now, if the SER has no ribosomes, this also means that the SER does not make proteins. It's not involved in protein synthesis. So, what does the SER do? The main functions of the SER is mostly to make lipids. It can make certain carbohydrates, and it makes our steroid hormones from cholesterol right here. Okay, the SER has some secondary functions that are really important. It can help with the detoxification of medications and harmful poisons. So it can make it so that a poison won't damage your cell. It will break them down, and it will help break down your medications. In fact, there's a lot of medications that are designed that they don't do anything until they start being metabolized or sent through your SER and broken down. And it's actually the piece that gets broken down by the SER that is actually going to start to treat whatever you're taking the medication for. Now we have a special type of SER called sarcoplasmic reticulum, and this is actually really important. We only find sarcoplasmic reticulum in muscle cells. Okay, so in muscle cells, the SER will store and metabolize calcium 2+ ions. So let's talk about this real quick. So as I said, muscle cells contain a specialized SER called the sarcoplasmic reticulum. Now, the main job of the sarcoplasmic reticulum is to store calcium 2+ ions in order to help your muscles expand and contract. In fact, if we didn't have calcium 2+ here, our muscles wouldn't work properly. Okay, so let's talk about what happens here. So as I said, the main job is to store calcium 2+, and that's because the release and reuptake of calcium 2+ ions is what's going to allow our muscles to function the way that they need to. Okay, so when we get a nerve impulse telling us to move a muscle, so we want to contract, so move it like this, that signal is going to tell our sarcoplasmic reticulum, let's just call it SR from now on, it's going to tell the SR to release stored calcium. Okay, so the release of calcium is going to allow me to contract my muscle. Okay, but what if I need to extend my muscle? Well, in order for our muscles to extend, our sarcoplasmic reticulum has to reuptake that calcium that it released. So when it reuptakes it, it's going to allow our muscles to relax and to extend. Okay, so SR is going to release stored calcium, which is going to cause our muscles to contract, and it's going to reuptake them, and the reuptake of calcium 2+ is going to allow them to relax and to extend. So pretty cool. But this is only this type of SER is only found in muscle cells. So the last organelle that we're going to talk about that's part of the endomembrane system is the Golgi apparatus. Now this is one of those organelles that's really weird because it's actually named after the scientist that discovered it, Camillo Golgi, if I said that right. He's an Italian scientist. So the Golgi apparatus is a lot like the endoplasmic reticulum. It's a series of stacked membrane sacs and tubules and vesicles that are going to make them up. Okay, so most of the stuff that comes out of the endoplasmic reticulum, so the proteins and the lipids and the steroid hormones that come out, are actually going to go to the Golgi apparatus for further processing. Now, I like to think of the Golgi apparatus as the post office of the cell. The Golgi apparatus is going to make sure that everything is shipshape, everything has the right modifications, and then it's going to put it into a vesicle and make sure it gets to where it needs to go. Okay, so post office. So a lot of things are going to get modified in the Golgi apparatus. Okay, this is what we mean by processing the proteins. And then everything, once it's modified in the way it needs to be, it's going to be put into a vesicle, a lot like the transport vesicle that came out of the endoplasmic reticulum. In fact, the transport vesicle from the endoplasmic reticulum actually travels to the Golgi apparatus and becomes part of its membrane system. And it's just going to dump its contents into the Golgi apparatus. So once that protein or that lipid has gone through all the modifications it needs, it's going to come to the end of the Golgi apparatus, and the Golgi apparatus is going to wrap it in another membrane and form another vesicle around it. Okay, and once this happens, we can ship or make sure that protein or that lipid gets to where it needs to be in the cell. Okay, some of them are going to be the digestive enzymes that get wrapped in the membrane that are going to become the lysosome in animal cells. Some of them are going to be vesicles that are going to then travel to the plasma membrane. In fact, most of the stuff that gets processed in the Golgi apparatus is actually destined to leave the cell. Okay, so most of the products from Golgi apparatus are actually going to be sent to the plasma membrane and not to anywhere else in the cell. Now, one of the ways that the Golgi apparatus makes sure that these vesicles get to where they needed is they're going to add a little carbohydrate tag, like a mailing label to each vesicle, that's going to help direct it to where it needs to go. Okay, so if our final destination is the plasma membrane, our vesicle is going to get made and it's going to travel along what we call the cytoskeleton, which we'll talk about after the Golgi apparatus, until it reaches the plasma membrane. So when it gets to the plasma membrane, it's actually going to incorporate itself into the plasma membrane. And while it does this, it's going to open up and it's going to dump all of its contents outside of the cell. Okay, this is how we transport things out of the cell, like our steroid hormones. Now, the Golgi apparatus has a receiving side. This is the side facing the endoplasmic reticulum. We call it the cis face. And the opposite side where things get shipped out is the trans face. Okay, so transport vesicles from the ER are going to fuse with the cis face and empty their contents into the lumen of the Golgi apparatus, like we talked about on the previous slide. And once those proteins and lipids are modified and have traveled through the Golgi apparatus, they are going to be packaged by the trans face and given their mailing label, that short chain of sugar molecules that's going to get it to where it needs to go after it leaves the Golgi apparatus. Okay, the last thing we're going to talk about in this chapter is the last organelle that we're going to talk about, and that is something called the cytoskeleton, so the skeleton of the cell. So the cytoskeleton of cells, of eukaryotic cells, are made from protein fibers. Now the cytoskeleton has several functions. It's going to help maintain the shape of the cell, a lot like the plasma membrane. It's going to hold organelles in specific positions. So the nucleus is going to usually be mostly at the center of the cell. Okay, so the nuclear envelope and the cytoskeleton are going to hold that into place. The cytoskeleton is going to allow for the movement of cytoplasm and vesicles within the cell. In fact, the vesicles that come out of the endoplasmic reticulum and come out of the Golgi apparatus are actually going to use the cytoskeleton kind of like a road system. Okay, so it's going to allow things to move. And the cytoskeleton within tissues, so cells in multicellular organisms, the cytoskeletons would allow cells to move within the multicellular organism. Okay, so the cytoskeleton is made of three components, so three different types of protein fibers. They are the microfilaments, the intermediate filaments, and the microtubules. Now, as I said, all these are protein fibers, but they differ in size, with the microfilaments being the smallest and the microtubules being the biggest. Now, all three components of the cytoskeleton are going to help maintain the shape of the cell, but their different sizes are going to allow them to do different functions besides maintaining the shape. So let's talk about each one. So let's talk about these specific parts of the cytoskeleton. Let's start with the smallest, the microfilaments. Microfilaments are made from the protein actin. Okay, so actin is a monomer and they're joined together to form the microfilaments, which is a polymer. Okay, so we have repeating actin molecules in our cytoskeleton. Now, these are really important. These microfilaments are going to help with contraction of muscles in multicellular species, so like us, but in single cell organisms, they're also going to help with cell movement, okay, and cell locomotion. But probably their most important job, other than helping to give the cell its shape and make the shape stable, is that it's going to help those vesicles move throughout the cell. So once things are done at the Golgi apparatus and we put them into those transport vesicles to go wherever they need to go, they're going to follow along the microfilaments to get to where they need to go. Okay, so the next part of the cytoskeleton we're going to talk about is the intermediate filaments. Now, the intermediate filaments are different than the microfilaments. They are larger. Okay, so microfilaments are about 3 to 5 nanometers in size, and intermediate filaments are about 8 to 10 nanometers. Okay, so they're about halfway between microfilaments and microtubules. Now, intermediate filaments are made from fibrous proteins like keratin. These proteins can be wound together kind of like ropes, which is why they get their name intermediate filaments. Okay, so we have these fibrous proteins. They are purely structural in function. They do not have any role in the movement of the cell, unlike microfilaments that do help the cell move, especially single-celled organisms. Intermediate filaments, they have a very specific job though. Their job is to maintain the cell structure by bearing tension. Okay, so they are going to be what takes all the stress added to the cell. Okay, so if they can't bear tension, the cell will actually shear in half. Okay, so by bearing the tension, they help maintain the cell shape, but they also have another job besides bearing the tension. They actually are going to act as an anchor for organelles like the nucleus, and they're going to keep them in place in the cell. Okay, so the last thing we are going to talk about are the microtubules. These are the largest structures in our cytoskeleton. Okay, our microtubules are made from a protein called tubulin. We actually have two types. We have alpha tubulin and beta tubulin. And in microtubules, they're going to repeat every other one. Okay, so alpha tubulin and beta tubulin are going to make what we call dimers, or two molecules bound together. So we're going to have 13 of these alpha tubulin and beta tubulin dimers, and they're going to be arranged in a circle that was going to make a hollow tube. Now, because the microtubules are made from tubulin, they're very rigid. Okay, so they're going to make the internal skeleton for some cells, and this is going to be a way to help the cell maintain their shape. But one of the biggest things is they're going to act like the road system for our vesicles. Okay, they're going to provide the framework for our motor proteins to move structures within the cell during cell division. In fact, the microtubules are going to be what moves things to opposite ends right before things divide. So if you have two sets of every organelle, one set has to be on one side of the cell and the other set needs to be on the other side. And microtubules are going to be what does that. Okay, so we're going to build our microtubules during that cell division. So one of the cool things about microtubules is that they don't just make up the internal cytoskeleton for cells. They actually make up a lot of the motility structures, so the structures that cells use to move. Specifically, eukaryotes have cilia and flagella. So the flagella work just like the flagella in prokaryotic cells. This is going to be those whip-like protrusions that are going to help the cell move. So in prokaryotic cells, they're going to rotate. In eukaryotic cells, they're going to beat. So they're going to wave back and forth. Okay. In fact, all eukaryotic cells have their own unique beat pattern. Now, cilia is going to be a lot like the pili in prokaryotic cells, and it's going to help with both movement and attachment. Now, the difference between cilia and flagella is their size. Flagella are longer, cilia is shorter. Okay, but they are both made from microtubules, and these microtubules are arranged in the same pattern. Okay, we have a 9 plus 2 arrangement, so we have 9 pairs of microtubules with a pair of microtubules on the inside. Okay, so all of our flagella and all of our cilia are made the same way. So this is what they're going to look like on the inside, which are going to allow for the movement of our eukaryotic cells. Okay, so let's recap because this is important. Both prokaryotic cells and eukaryotic cells have flagella. They just move differently. Prokaryotic flagella rotate, eukaryotic flagella beat. Cilia and pili are not the same thing. Okay, pili is only found in prokaryotes. Cilia is only found in eukaryotic cells. It's found mostly in animal cells. So that is all our eukaryotic structures. We will talk more about plants versus animal cells and the remaining few structures in your worksheet in the next lecture.